Good morning, everyone. So Lata and Albert have really sort of set the stage for me, and I'm going to talk about something slightly different, where I'm not a PD expert. So I was instead going to give you an overview of how population genetics can sort of really help us get insights into the molecular etiology and the mechanisms underlying Parkinson's disease. So this is familiar to all of you, but sort of from a geneticist's viewpoint, we traditionally classify Parkinson's into familial and sporadic, with familial being a very small percentage of Parkinson's cases. And traditionally, for the last 40, 50 years, we've used pedigrees and linkage studies on these familial cases to identify the causal genes for Parkinson's. So right now we have a small but sort of consistent list of genes that keeps popping up every time we have these cases of Parkinson's, of familial Parkinson's disease. On the other hand, for the 85-90% of sporadic cases where we do not have a family history, studies have been more recent, and there have been, over the last 20-ish years, these population-based studies. Initially, they were genome-wide association studies that used microarrays, where you look at common variation across large populations and compare the variants seen in cases versus controls to identify these variants associated with Parkinson's disease. Now, with sequencing becoming cheaper and more common, these population-based studies are now sort of shifting towards sequencing, where we can identify both common and rare variants that together lead to your increased risk of PD. One thing I want to highlight is that even though geneticists and often clinicians we separate sporadic versus familial, and we sort of think about risk factors versus Mendelian variants, in terms of the genes that we identify, we've identified 90 risk loci from these population-based studies, but there's a significant convergence of these 90 risk loci with the genes identified from familial-based studies. So there is a convergence of molecular mechanism, even though we sort of think of them as familial and non-familial, at the molecular level, there does seem to be a significant convergence. And thus, there is a benefit to be gained by looking at these population-based studies. One example of that I'll illustrate at the MAPT locus. So from all of you are familiar with sort of the familial studies showing very, very strong missense or splice mutations in this gene that are associated with high risk of PD. One of the strongest GWAS signals from the first GWAS of Parkinson's was a SNP, a single nucleotide variant, at the MAPT locus. And a bunch of researchers at Columbia University sort of went in and dug deeper into that locus to ask, how is this variant affecting, how is this signal at or close to the MAPT locus affecting PD? And what they saw, I'm going to use the pointer. Yeah, what they saw here is that the having different genotypes or having different alleles at this one particular disease-associated position in the genome led to different levels of the inclusion of exon 3 of the MAPT gene. So splicing, this is a splicing variant that has been associated with PD, and sort of gives us another potential mechanism by which a variation or genetic variation at this locus can lead to PD. So from familial studies, it was primarily exon 10, that splicing at exon 10 that was associated with PD, but sort of adding these population-based studies gives us more mechanisms by which that connect the gene with the phenotype. Similarly, at the SNCA locus, this was another one of those top hits that came up from the GWAS, and researchers really went and dug deep to figure out what's going on. So the plot on the left, the y-axis just shows the significance, the p-value, the significance of the association, and across this window on chromosome 4, and you see this one signal really stand out. They saw, okay, so there is this one variant that is really associated with near SNCA that's associated with PD. Is that it? So what they did was they took out this signal and conditioned on this signal and looked for additional signals in that same region. And when you do that, you actually see a second signal that is also significant. So those p-values are 10 raised to minus 10. So like really quite strong hits, a second strong hit in that same region around the SNCA gene. They now said, okay, is there anything else? They conditioned on both of these and saw a third significant signal. So there seem to be essentially 3 independent genetic signals and that same gene, all of which seem to be additively or perhaps non-additively contributing to the risk of PD together. They haven't really dug deeper into the second and the third signal yet, but that first signal, which is around the 3' end of SNCA, they overlaid it with other kinds of genetic functional genomics data. So here what they did was that SNP RS356182 is that significant SNP that I had shown you in the previous slide. When you overlay it with functional genomics data, open chromatin, whether it is in a transcription factor binding site, whether it is found in an active enhancer marker in the genome, you find that the SNP very nicely sort of overlaps with a bunch of active regulatory marks in the genome. And they also then later did an RNA-seq to show that this SNP is associated with levels of one particular transcript of SNCA. So even though it's in 3', we usually think of regulatory variants as being 5', but this seems to be an example of a 3' variant that is regulating the expression of one particular transcript of the SNCA gene. Another sort of big theme in these population genetic studies for the last few years has been population heterogeneity. So one theme in human genetics has been that most studies are done on European populations, and what does that mean when you try and extrapolate these results to other populations? So the first GWAS on PD in African populations was done last year actually, and here you see that if you compare the results of the European GWAS at the bottom with African GWAS at the top, you see this one very strong signal pop up in African populations that you don't see in the European population. This is at the GBA1 locus. It's not because this variant is not leading to PD in European populations, but that this variant was just not present, right? Different populations carry different variants that are enriched in those populations, or perhaps in an extreme case even unique to those populations, and therefore discovery in one population doesn't necessarily extrapolate to discovery in another population. So an increasing theme in the PD genetics community has been to increase this diversity. How can we make Parkinson's disease genetics equitable so that the clinical and the medical benefits from it can be applied throughout the globe? In fact, the same group also sort of looked at those 90 risk loci associated with PD, and they looked at the effect sizes across different populations on the y-axis. The blue sort of shows that the effect sizes are consistent, and the less blue it is, the less consistent they are across populations. And sort of what jumps out at you is that's quite a bit of non-blue signals you find, right? Which again really points to how the results we see from European-based studies may have to be treated with a pinch of salt when you try and extrapolate them to other studies. And this might hold true in the context of India as well, though sort of it's unlikely that the difference between European and Indian populations are going to be as much as the difference between European and, let's say, African populations. So another big theme in the population genetics community has been the use of what are called genetic risk scores. So if you combine your genetic risk across the genome into a single score, can that be used as a biomarker or as an early predictor of PD? People have been very excited about this because it can be, you can get your polygenic risk score at birth, right? You don't have to wait for any symptoms for your risk score to be calculated. And an early estimation of this risk score can really help you, let's say, with early interventions, lifestyle changes, and so on. This has been it's sort of becoming mainstream in the field of cardiovascular genetics, right? Where sort of it's actually becoming a commercial, it's getting into the commercial realm primarily, and there was this one landmark paper in 2018 that led to that. So the two plots here are from this paper by Amit Khera in 2018, where they showed, if you look at the second plot on the right, if you sort individuals by their genetic risk score for heart disease, right? Only based on genetics, you're sorting individuals into percentiles. So these are individuals with the lowest genetic risk for heart disease, and these are individuals with the highest genetic risk for heart disease. And then on the y-axis, what you're plotting is how many of them actually got a heart attack in the time span that they were studied for. And you can see this huge difference, this sort of almost 7 or 9-fold difference, I think, between individuals in the lowest percentile and individuals in that highest percentile, showing that your genetic risk can accurately, to a large degree, predict the risk for increased heart disease in the extremes of the percentiles. So increasingly, this has been used by in the commercial space. There are kits where you can send your DNA and get your genetic risk score for heart disease. And this is sort of one of the branches that human genetics is hoping to help with personalized medicine, right? Can you actually give you a personalized risk score for any disease? For PD, we are not quite here yet, and I think this is where one of the challenges lies. Compared to heart disease, PD does have less heritability. There is less of a genetic component to it, so it may be a more difficult, a difficult challenge in the case of PD. But so far, the polygenic risk score for PD gives us an odds ratio of about 6 for that highest percentile compared to the lowest percentile. So there is a hint of potentially something there, but we're nowhere close to that cardiovascular the success of polygenic risk scores in the cardiovascular space. But this is one potential area of that could be very exciting for early biomarkers and early risk prediction. I've sort of so far spoken about these common variants as separate from monogenic Mendelian risk variants, but in an individual, I mean, biology doesn't care. The combined risk of an individual is just the combined risk from both rare, your monogenic variants, and your common variants. And in fact, increasingly with sequencing being more and more common, with these population-based studies also using sequencing instead of microarrays, we're beginning to ask these questions about how do these common and rare genetic factors, what is that interplay between them? How do they affect each other? And so especially some questions that are being asked is, the question of incomplete penetrance is one that, as clinicians, face a lot. You might have that high confidence risk variant, but it's a control. You show no symptoms. Why is that? Can common genetic risk help understand that or stratify patients a little bit better? And another question is, how do common variants modify the penetrance of a rare variant? Right? So this was a study that showed that there is some promise here as well, where the authors, this is the International Parkinson's Disease Genetics Consortium, they looked at patients with Gaucher's disease and really separated them with Gaucher's disease without PD and Gaucher's disease with PD. And on the y-axis, what they've stratified them is the polygenic risk score for Parkinson's disease, right? And what you see is that people with Gaucher's disease but do not have PD have a lower polygenic risk for PD than patients with Gaucher's disease but that also have Parkinson's disease. It might seem like that's a fairly straightforward result. I mean, yes, of course, they have PD, so they have a higher genetic risk. But these sort of quantifications can potentially help us understand or predict the onset of symptoms in a set of individuals with, let's say, atypical Parkinsonians or that belong to the family of Parkinson's disease conditions. Another sort of, a similar sort of, in that same vein, we're sort of trying to understand how these common and rare diseases, common and rare variants act together, right? And we often think about that in the context of what's called the liability threshold model, where you're assuming that, let's say this is a distribution of risk, and that risk can be a genetic risk plus environmental risk, everything, all kinds of risks thrown together. And let's say there's a distribution in the population, and anyone crossing this red threshold has the disease. Now, that risk, the cumulative risk could be contributed to by environment, by one big large effect monogenic variant, or by multiple common variants with small effects. And the question here is, if a diagnosed patient, or if a patient has one large effect monogenic variant, they are likely to be diagnosed. Whereas if a person with PD does not have that single large effect genetic variant, they may have many, many small common effect variants, they are more likely to be undiagnosed. So can we sort of compare the genetic risks of diagnosed versus undiagnosed patients and see what we find? This was done for neurodevelopmental disorders, where they, I'll skip this because I'm running a little late, but this more recently in a paper by some of the researchers in this room, they actually performed this analysis for an Indian cohort of 600 or so individuals, where they compared the last two panels. They sort of compared PD cases with a high confidence pathogenic variant with those without that high confidence pathogenic variant. And they saw that if you have that high confidence pathogenic variant, you're likely to have a lower genetic risk score from your background, from your genetic background. So that liability threshold model does seem to add up where, and this sort of, this is a motivation for us to start considering genetic background and common variant risk in our assessment of PD. I will quickly stop and just say that there are, the main point is that we do need to have more of these in India, more population-based studies in India, and there are quite a few by people in this room. And we're also hoping to contribute to these efforts through a funded project at CBR as well, in collaboration with NIMHANS and funded by the SCAN project. Our goal is to sequence 1,000 cases with young and late onset Parkinson's disease, and we're hoping to longitudinally follow them up to look for genetic susceptibilities to PD in our populations, and also to look at stratifying them by different degrees of severity. I'd like to sort of stop here and take any questions. Thank you.
Thank you, Dr. Shweta, for an interesting talk. Any questions?
Nice talk. Very naive question. Somewhere you mentioned that Caucasian versus Africans and Caucasian versus Indians may be different, may not be the same, and we may be more closer to the Caucasians, if I understood right.
Yeah.
Can you just shed light on that?
Yeah. I did run over that very quickly. I think what I meant was that genetically, if you plot just genetic distances between different populations, we are much closer to European populations than, let's say, Africans are. The most, distinct population is the African population, so you would expect more differences. They contain way more variants that are just not seen anywhere else in any other continent, whereas that's not so true for our population. Yeah.
I think the way you pointed out that a lot of genes are not found in the Indian populations or subgroup. So we are noticing that a lot of VUSs are coming up.
Yeah.
And I think that is where the research is stopping for us. We need to have a lot of functional studies are required to understand what is the importance of all those things. How well we are equipped to do those type of studies. That's the question. I thought if you have any answer on that, you can let me know.
Yeah, I mean, I think that is the challenge we are facing as well. With our work, I think what you have actually three classes, not monogenic, not diagnosed versus not, but it's diagnosed, VUS, which is this huge list, and then not, right? And yeah, I think I don't have an easy answer. I wish I did. But I wonder if we can sort of incorporate these statistical methods to try and maybe understand which subset of these VUS are more likely to be true causal versus not. That's one thing. Another thing is a lot of these VUS can potentially be resolved once we have better population-based reference panels. Once we know which variants are more common, are found in healthy populations versus not. So I think that's the second branch, which will help, at least in the context of India. Third, I wonder if there need to be more these high-throughput molecular screening techniques to try and just predict the function of these VUS. I think efforts are ongoing, but for some reason they've not been able to assist much with this problem yet. But that would, I think, be very helpful. But if anyone else has thoughts on that, I think that's the challenge of our times, and it'll be helpful to have a discussion.
I think the primary point was that only because at this point because I'm part of the Global Parkinson's Genetics Program, and so people want to fund for us for PhD programs. We want many people to take up these things for a lot of things which are unanswered, especially when it comes to Indian subcontext. A lot of work is not done at all. There are opportunities are there, funding is there, and someone has to apply. That's the whole. Because for us, the clinician parts, what we are sitting, we are looking at. We can have sample, we can have clinical data. So all that thing is there, which is in abundance in India. That is not an issue at all. But the point is, after that, what is the question? That is where we are stopping now. There, I think probably. If some of you take that lead down there and any help is required, definitely Ravi's are there, I am here, and Shweta is there, and we can help you to guide out to get the right resources so that your career path is also struck in that direction for a long time to go on as such. That's the whole goal here. Thank you.
Any other questions?
Are there any leads, initial studies, GWAS studies, any lead, any LOSA where we're looking at trend in our population, whether it is GBA or LARC, any trend initially?
I think the only paper I'm aware of is the one by PRAI that came out this year, I think. they found, they didn't find, if I remember correctly, they didn't find any novel variants, but they replicated existing signals in European populations at LRRK2, and I think it was GBA, or it was either GBA or SNCA, I can't remember, one of these two genes. So it was, they showed a high degree of consistency with European signals, but I suspect that with larger sample sizes, we're going to find these novel signals that, and especially my hope is that all of these different consortia also come together so we can run a larger, more powerful study than multiple small studies, and that will perhaps help us really find these novel signals.
Yeah, so I just wanted to comment.
Hello.
I was
Yeah.
part of this
Okay.
study, So
Hello.
coming to Dr. Prashant's question, a lot of viewers, we are also facing challenges, diagnostic companies. So one example that we show in this paper is the GBA variant that is previously a VUS or a null variant. We found new evidences with the in vitro validations and all. Probably that will transfer to pathogenic or likely pathogenic in the PD diagnosis. So likewise, such studies are needed in large sample size to make more, more diagnostic yield, which is important for companies like us as well. Okay, thank you.
I think this is a universal problem. As sequencing samples are going on and we're identifying variants and interesting genes, what is also required from the research community is to, the experimental research community is to look for some kind of a high throughput system, a brute force method. Individual researcher works on one gene, one variant, one mutation, probably sometimes their entire life to figure out what it does. So at the end, you could still have a consortium of scientists, and it has to be engaging nowadays, making transgenic, let's say C. elegans or flies, but certainly not mice at that level is not going to be possible. But one can do those, and you can see if it recapitulates at least some aspects of the disease phenotype. And then you need to do rescue, either genetic rescue or available pharmacological rescue of available FDA-approved drugs or treatment modalities. And if that is happening in a pipeline fashion, it's not very intellectually stimulating process. It initially for scientists, so nobody's going to pick it up, but some of us have to come together to get that thing going.
Hello. Can you highlight me about the DNA mapping? Will give you a different idea of the genetic differentiation between the other type of this one and the Parkinson disease. This will give you a good classification.
Oh.
So has it been done? Can you highlight me about it? I'm a person affected by the PD, so I would like to know whether such a mapping has been done, DNA mapping of the different regions of the world, so that how the pattern goes.
So,
What
yeah.
are the symptoms and indications?
Yeah.
Thank you.
Okay. Yeah, so if I understood correctly, the question was how much are there genetics mappings that's been done to find out why you have different kinds of symptoms,
Yes.
why you have PD and why you have different kinds of symptoms. Yeah, so that's sort of those questions are what a lot of these studies are trying to dig deeper into. There have been a lot of studies where for some very well-known mutations, we have a good sense for what is the risk for PD, for Parkinson's, and what kinds of symptoms they tend to be associated with. So there's a small subset of mutations for which we have a reasonable idea, but the challenge is that these explain a very small fraction of the cases. So there are a lot of people, in fact, most people for which we don't have that mapping done, and that's what we're hoping to do with these studies. And that's I think a shared goal for a lot of people in this room.